have Dr. Stephanie Leal as our speaker today. So Stephanie is an assistant professor here at the Department of Integrative Biology and Physiology and the Department of Psychology. It's joint appointment at UCLA. She also holds the title of an adjunct assistant professor at Rice University. And so at UCLA, she directs the neuroscience of memory, mood, and the aging lab. So she received a PhD from Johns Hopkins in brain sciences with Michael Lyasa as her advisor. She then did a postdoc at Berkeley with Bill Yegus, and then she spent a few years at Rice as a faculty before getting recruited here to UCLA. So she's won several awards, including the NARSAD Young Investigator Award, the Bright Focus Alzheimer's Disease Research Foundation Award, and the Alzheimer's Association Award for Young Scientists. So the lab here is focused on understanding the neurobiological mechanisms underlying episodic memory for aging and disease.
Okay, so my lab is focused on this primary research question, which is very broad. Why do we remember certain experiences over others? So what makes our brains, we can't take everything in that we experience in the world. So how does our brain pick and choose what information is going to be stored for long-term use and what is okay to forget? Or maybe the system goes, awry sometimes. So we're going to talk about that in two situations of depression and aging. And so my lab tackles this question, thinking about three different levels. So first, how can we best measure memory? So lots of studies use different types of stimuli, including as simple as word stimuli, like this word baby, maybe getting more complex with the line drawing of a baby. Object stimuli are commonly used in human studies of memory. Scene images, again, a little bit more complexity here. And then you can see all the way to this end of the spectrum in terms of a video stimulus that is a little bit more rich, and you can see emotion and get a little bit better sense of that experience compared to just seeing the word baby, right? So we think about this in terms of trying to measure memory in a more naturalistic way as we think about how we can measure why we're remembering certain experiences. We also think about individual differences a lot in my lab. So if you think back to all of your experiences throughout your life that you can recall, we all have a very unique path and what we remember in that moment about ourselves, about our families, our semantic memory, right? Memory for things that we just know about. And so where we are today is very different than where we were 5 years ago. And so thinking about how all of our experiences make us who we are and how that might bias what we remember and forget in the moment is something we like to think about in the lab, and how that contributes to the development of disease. And then finally, we're also interested in thinking about modulating that memory. So if memory goes awry in conditions such as depression and aging and Alzheimer's, can we change and modify those memories such that maybe we don't have this negativity bias in memory that we see in depression and age-related cognitive decline? If there's a way we can rescue that and intervene early on. So we think about this in a lot of different ways. So we use music stimuli, emotion regulation, antidepressants, lots of different ways that we could think about modifying our memories. So just sort of a broad overview of how we tackle those questions in my lab. We develop and utilize sensitive cognitive tests. In particular, we'll talk about a mnemonic discrimination task that taps into hippocampal pattern separation. That's really kind of one of the core stimuli and tasks that we use in the lab. We pair that with advanced imaging methods. So we use high resolution MRI, both structural and functional imaging, as well as PET imaging of amyloid and tau pathology in the context of aging. And we're also more recently utilizing MRS techniques, as well as really getting to the nitty gritty of certain regions of the brain, like the locus ceruleus, for example. And we take those two aspects of the lab and we try and apply those in different types of intervention settings. So for example, as I mentioned already, aging and Alzheimer's disease is one big area we look at, depression, and then different types of modifications of trying to intervene when memory goes awry. So today I'm going to be talking about a little bit of background on the emotional modulation of memory. This is sort of the approach we take in thinking about how we remember significant events, things that are important and have emotional significance. Then I'll talk about when memory goes wrong, situations like depression and aging, where we see biases and changes in memory and what people remember. And then I'll touch back on some ongoing and future directions related to those areas of the lab I brought up at the beginning. All right, so in terms of the emotional modulation of memory, in particular, we're really interested in episodic memory, or memory for experiences in our lives. And the classic definition of this that was proposed by Tolving is episodic memory is made up of these what, where, and when components. So what happened, where did it happen, and when did it happen? And lots of studies tackle these independently, as well as sometimes, joined together, so spatiotemporal information. But a lot of studies really kind of focus in on one aspect of these different parts of episodic memory. And we were really interested in what we say is a missing W in this what, where, and when definition, and that's the why. So kind of this broader contextual information of why are we remembering the what, where, and the when. So thinking about, again, that individual person and what they've experienced that makes up why you're remembering certain information. This was a review paper that we published last year in Cognitive, Affective, and Behavioral Neuroscience, led by Fernanda, who is graduating this spring somehow. And this article won Best Article in Cabin, so we were very excited about that. And so if you haven't checked it out, I highly recommend giving it a read and kind of broadening and thinking about episodic memory in this way of providing this why component. This is a component we try to focus on in the lab. And the reason, again, is thinking about what we are remembering. And so we need to remember significant experiences from our lives in order to be able to use that information to guide future behavior. And so, we argue that this is a really important feature of episodic memory that's often understudied or sort of ignored a lot of times. And I like this quote by William James. He says that some events are so emotional as to leave a scar upon the cerebral tissues. So these types of events are literally changing the brain and the structure and how different brain regions are talking. So how does our brain do this? How are we able to prioritize what information we're going to remember? And so to get into that, we're going to focus on the medial temporal lobe. I'm just going to highlight some of the regions that we focus on in the lab. So the hippocampus, which is that green structure in the middle there, very important for episodic memory. And we're going to talk about some of the computations that it performs in just a minute. And then we have the entorhinal cortex, which is this reddish structure right adjacent to the hippocampus. That's the main input into the hippocampus. And then we have the amygdala here in pink, which is often thought of as a fear center or emotion, but I like to think of it, and others as well, as really noting biological significance. So things that are important, whether it's positive or negative, it's kind of signaling this is something important, this is a significant event. And so we can measure the amygdala, and in particular, the basolateral amygdala is really the subnuclei of the amygdala that's involved in modulating hippocampal function. And we can measure different subnuclei of the amygdala. There's been lots of different protocols developed using high resolution imaging to be able to tease apart different parts of the amygdala. And then same for the hippocampus. We're able to look at subfields of the hippocampus using high resolution imaging, both functionally and structurally. And so we can draw these different ROIs in both of these areas and look at activity in those brain regions within the medial temporal lobe as it relates to this emotional modulation of memory and how the amygdala and hippocampus are interacting. So if you think about an emotional experience from your past, take, for example, this picture from my wedding in 2018. For me, this might be a very emotionally significant event. As many of us, who are married, that might be one of these more positive experiences that you recall from your life. However, if you think about somebody who attended my wedding, this is my mom's friend Terry, it might not be as emotionally salient for her as it is for me. So she might have a very different recollection and memory of this event compared to my memory or my husband's memory. She also attended 4 other weddings this summer. And so you can imagine there was a lot of interference in those events. Weddings often follow a very similar timeline, the ceremony, dancing, cake, all kind of the same stuff. And how do you remember differences from one event to the next? So you can even see from here, there's very similar flowers, similar dress, all that kind of stuff. There's a lot of overlap and interference in her memory, especially of these 4 different weddings she attended. And this is something we deal with in our everyday lives all the time. For example, every single day you might drive to work, take the same route, park in the same parking structure, and you want to be able to remember where you parked your car one day to the next. So this is a really important feature of our episodic memory, and that's being able to reduce interference across similar experiences so that we can recall unique events, which is really kind of the key feature and function of episodic memory. Also, I last year in 2024 now had twins, identical twin boys. And so I sort of have my own everyday example of interference in trying to, tell apart my twin boys. I can tell them apart pretty well, so I have developed expertise and I'm able to notice subtle differences. But for people who meet them for the first time, often struggle telling them apart. And so for our brains to be able to do this and recognize, my identical twin boys as different people, or, separating out experiences that have a lot of overlapping content, computational models have proposed that there's 2 key computations that can help with reducing interference in some cases. And so the first computation I'll talk about is called pattern separation. And that's this ability to reduce interference when there is overlapping information. So take, for example, A and A prime. Let's say those are my twins, Hayden and Harvey. We want to be able to recognize them as different people, since they are different people, even though they look similar and they have overlapping features. And this depends on the dentate gyrus subregion of the hippocampus. This region is, I won't get into the details, but it's kind of really built to have sparse coding that allows for unique representations to be stored. And it can then send that information down to the CA3 subregion, out through CA1 to the rest of the brain through this trisynaptic circuit, and separate out these individuals as different people. On the flip side to that is pattern completion. And so this is when, in the face of interference in this A and A prime example, sometimes we don't want to actually separate them out. We want to generalize and say, for example, this is my dog Dexter. It's the same dog, but he looks a little bit different in both of these images. And so you want to be able to recognize that that's the same dog, even though he looks a little bit different in these 2 pictures. And this depends on the CA3 subregion of the hippocampus in being able to perform this computation. So taking some information that's overlapping and completing that pattern and recognizing something that you have seen before, even if it looks a little different. And importantly, for our high resolution imaging, we're not able to tease these apart. And so we will, for the data I'll be presenting, we combine the dentate gyrus and the CA3 subregions together. And we're looking at activity in the brain, and I'll talk about the task in a minute. But during, when people are actually discriminating similar experiences, like, for example, the twins, being able to, if they're doing that successfully, then we're assuming that this pattern is being propagated down the hippocampus. So interpretation is a little muddy, gets a little muddy there. But this is, I just wanted to give you a heads up on that we are going to be looking at these regions together when people are actually performing these types of pattern separation computations. Okay. And so tasks in humans have been developed to try and push our memory systems to the limit by inducing interference in memory. And so if you look at this first panel here in A, this is the classic object version of the task where participants are shown a series of object stimuli, and then they're tested on their memory for those images. And you can see that some are exactly the same, like this lobster, and some are brand new, and then some are similar, but not exactly the same. And so like, for example, this apple looks very similar to the one they saw originally, and same with the toaster and this blue ribbon here. So those images that look very similar, but they're not exactly the same, are meant to induce this interference in our memories and say, did I see that toaster before? Or is it different in some way? And they've created versions in the spatial domain and the temporal domain as well. So sort of testing these different features of the what, where, and when of episodic memory. However, we wanted to, again, contextualize this a bit more and think about the why. And so we developed an emotional version of these tasks. So we used a little bit richer of stimuli. We used scene images. And we showed participants neutral, negative, and positive images during encoding. And then after a delay in this study, either immediately or 24 hours later, we tested participants' memory for those images. And they could see a repeated image, just like a standard memory test. Have they seen this before, yes or no? We show them brand new images they haven't seen before. And then we show them these lure images, which are those similar but not exactly the same images that tax this interference and pattern separation ability. And so we have two measures that we extract from this. One is a general memory measure, target recognition. So just, do you remember the same image you saw before? And then we also calculate a lure discrimination index, where we look at the ability to discriminate that is actually a different funeral scene than one they saw before. And we'll be using this task throughout today's talk. So if anyone has any questions, let me know. Yeah.
So half of the images that they learn are immediately, and the other half is the next day.
So for this version of it, it was two separate groups of people. So one group got tested immediately, another group got tested on all of the same images 24 hours later. But we have done that version too with other tasks. But yeah, so this one is two separate tests, different people, and these are the results from that. So we find, this was work that I conducted during my graduate work, where we found that target recognition, when we look at negative and positive memory compared to neutral, immediate performance is in green and the delay is in blue. And you can see that there's very little forgetting of the emotional information compared to neutral, which is what you might expect when you think about emotional memory. We want to remember these things better. However, for lure discrimination, we actually find the opposite to be true. So there's more forgetting of emotional details compared to neutral. So there's this gist versus detail trade-off that we find evidence for. It has been found using other types of paradigms. And we kind of replicated that here with this type of paradigm in thinking about gist of emotional experiences being more preserved at the expense of emotional details. And so we think that it might be adaptive in cases where there's emotional arousal, very, potentially stressful or high arousal situations, where it might be better to generalize in those situations rather than focus on details that might not matter as much. And when we look at the brain during the performance of that same task, we find signals consistent with pattern separation, and especially for emotional information. So the white bars here are negative stimuli and the black bars are neutral. And you can see that during correct rejections of those lure images, so like the funeral scene, for example, participants showed greater activity in the dentate gyrus CA3 subregions during negative lure correct rejections compared to neutral, as well as relative to false alarms when they got those wrong and they were generalizing in those situations. And then when we look at the amygdala, we find that it's just generally, regardless of if they're getting them right or wrong, showing greater activity during negative images. So no matter if they're correctly rejecting or false alarm, the amygdala is sort of just signaling emotion. It's greater activity for negative images compared to neutral. So it seems like the amygdala and the hippocampus are working in concert with one another to support emotional pattern separation, where the amygdala is sending this emotion signal and the hippocampus is sort of integrating that in, but more selectively, in terms of memory performance during these lure correct rejections. Yeah, question? Good question. It was a little more complicated with the positive stimuli, so that ended up going into the supplement. But I will talk about positive for the rest of the stuff today. So yeah, it was a little more messy. It was definitely stronger for negative, and our images, like many others, tend to be more arousing for the negative images compared to positive. Our positive images are still more arousing than neutral, but not quite as arousing as negative images. So we do tend to see more consistent, strong signals with negative typically. Yeah, great question. Okay, so that sort of set the basis of thinking about these computations and how the brain might be supporting emotional modulation of memory. And so we wanted to take this into a condition where we know there are memory deficits as well as memory biases in terms of emotion. So in depression, there's often just general episodic memory deficits, which we did find evidence of across both of those measures. So target recognition and lure discrimination, we find just worse memory for neutral images. But we also found evidence of a negativity bias where depressed individuals tended to perform better on this negative lure discrimination measure. We didn't see evidence of it for target recognition, but we did for these details in these images, for example, like that funeral scene that I've been going back to. So they tended to be able to discriminate those images better than healthy controls. And when we look at the brain in the same regions that we were just talking about, in the amygdala and the hippocampus and the dentate CA3 subregions, we actually find a shift in dynamics here, where there's actually decreased dentate gyrus CA3 activity and hyperactivity in the amygdala. And so, you know, this kind of abnormal shift in these activities that were associated with depressive symptoms really got us thinking about how is it possible to normalize these signals in some way and rescue the memory deficits and this negativity bias that we see. And so it seems like the amygdala being hyperactive is really driving some of what we see in terms of that negative hyperdiscrimination. So we wanted to next turn to, again, that modification piece of can we change both the cognitive profiles and the neurobiological profiles with whatever types of interventions that are out there. And so the first place we turned was antidepressants. We also have studies on emotion regulation where we find very similar findings. But for the sake of time, I'm just going to talk about our work on antidepressants in terms of what do they do to these cognitive and neurobiological profiles. So a little background on antidepressants. Rodent models have shown that there's increased neurogenesis in the dentate gyrus subfield of the hippocampus. And so new neurons are growing there when antidepressants are being taken. And also that's been associated with rescuing memory deficits, and especially on tasks that have interference, like these pattern separation type tasks. And this is all in animal studies. And also, if individuals are taking SSRIs, there's been associated with larger dentate gyrus volumes compared to untreated depression in humans. However, a really big caveat with individuals taking antidepressants is they fail about 50% of the time. So people aren't necessarily going to be responsive to taking antidepressants when they have depression and depressive symptoms. So this is a big problem, as you can imagine, in treatment. Another complicating factor with antidepressants is there's lots of different types of antidepressants. So the most commonly prescribed first-line treatment are SSRIs, which target serotonin primarily. And then if those don't work, physicians will often prescribe other types, which we call multi-action, which target multiple neurotransmitters, so serotonin and norepinephrine, or norepinephrine and dopamine, like SNRIs and NDRIs. There's lots of other ones as well. And there's also another category of polypharmacy where physicians will prescribe multiple antidepressants as well. So it gets very complicated very quickly. And what neurotransmitters they're targeting, we think is really important, and how that might modify what we see cognitively and what we see impacting the medial temporal lobe. The medial temporal lobe is very densely modulated by all of these neurotransmitters. And so we sort of wanted to just take a first stab at this question and look to see whether people taking antidepressants already show any sort of differences on emotional mnemonic discrimination, as well as underlying neurobiological function with our high-resolution imaging measures. So Madeline Castro, who's a fourth-year PhD in the lab, and an undergrad at the time, Taylor Phillips, who's now in med school, conducted this study in young adults ages 18 to 34. And we were just recruiting people taking any type of antidepressant. We didn't care. And we ended up getting 28 people taking SSRIs and 20 taking other types that we sort of lumped together and looked at. And so as expected, most people were taking SSRIs because that is the most commonly prescribed antidepressant. And we measured target recognition and lure discrimination on that emotional pattern separation task. And then we also collected a host of other measures, including Beck Depression Inventory to measure their current symptomology, antidepressant history, neuropsychiatric diagnoses, as well as a perceived efficacy score of their antidepressant. So this is a pretty crude measure that we started with in this study, where we had a scale 1 to 10, and people rated how they felt before starting taking antidepressants on a scale 1 to 10, 1 being not depressed, 10 being severely depressed. And so they would rate themselves for how they felt before. So it was a retrospective measure. And then they rated how they felt in that moment, after having been taking antidepressants. And so we performed a median split on this measure and looked at individuals who were responders, who showed greater reported retrospective change, and non-responders who showed less of a difference here in terms of how they rated themselves before and after taking antidepressants. So what we found, if we look at target recognition, there were no differences between responders in blue and in non-responders in orange. However, when we looked at lure discrimination, we found that there was a reduction in the negativity bias in individuals who were responders versus non-responders. And we saw this enhancement in neutral lure discrimination. So the exact dysfunction that we saw in individuals with depressive symptoms, it was completely reversed when you compare responders to non-responders. So this is really exciting. And the fact that it was selective to lure discrimination was really interesting and suggests that we might be missing something when we're just looking at general memory, and that it might be more selective to particular subfields that are really impacted with antidepressant use and in terms of alleviation of symptoms. So when people report that their antidepressant is effective for them, and retrospectively, it seems that these individuals show this reduced or reduction in that negativity bias and enhanced neutral memory. So kind of rescuing those deficits that we saw before. So then we turn to the brain. And so Maddie continued this line of work, but with high resolution imaging, to look at whether or not there were differences in responders and non-responders, as well as depending on the type of antidepressant in medial temporal lobe activity. So mainly the dentate gyrus and CA1 subfields we focused on, as well as in the amygdala, the basolateral amygdala, and the central nucleus of the amygdala. And so for this study, very similar recruitment strategy here. We wanted to get individual types of antidepressant groups large enough, but unfortunately we didn't. So for the SSRIs, we got a lot of people, 37 individuals taking SSRIs, which are single action antidepressants. So they are targeting serotonin. And then we've collected some people taking SNRIs, NDRIs, and multiple that we sort of, again, lumped together here into this multi-action group that are targeting multiple neurotransmitters. And we also collected some control data as well from depressed, unmedicated, and healthy controls. And again, this was ages 18 to 35, and we excluded any we didn't exclude them being diagnosed with other things because there's lots of comorbidities when you have depression. You probably will also have anxiety and other things. But we excluded anyone taking other types of drugs. So that was the primary exclusionary criteria. And then we got the same measures that we did before with the emotional mnemonic discrimination task performance, as well as history of antidepressant use and all of that. But we also in this study sort of got a little bit better of a retrospective measure of how they were feeling before taking antidepressants and gave them the Beck Depression Inventory as a retrospective measure. So how did you filled out the Beck Depression Inventory as if it was before starting the antidepressants, and then they're filling it out for how they're feeling that day. And so we took a percent change score here. And so this is kind of more in line with how clinical studies do this. So if they reported a greater than 50% change, they were called responders. If it was less than 50% change, they were called non-responders. So jumping right into the data, we looked at activity during those lure correct rejection trials, where we expect these signals of hippocampal pattern separation to be present. And we found that, and let me just orient you to the colors here. So the reddish bars are non-responders and the blue bars are responders. And the darker bars are the dentate CA3 and the lighter bars are CA1. And so you can kind of tell most of the action is going on here in the negative stimuli, where we found that the non-responders were showing this hyperactivity relative to non-responders, specifically during negative lure correct rejections. And when we looked at the type of antidepressant they were taking in terms of these broad categories of single action and multi-action, we actually find an interesting double dissociation here, where if you're taking SSRIs, you show greater activity in the dentate CA3 compared to CA1. And then that's actually reversed in those taking multi-action antidepressants. So we actually see greater CA1 activity relative to dentate CA3 activity. So there seems to be something going on, and these effects were in the non-responders. So it seems that non-response to antidepressants isn't simply just not improving things, but it might be changing these medial temporal lobe subregions and their activity. And it may depend on those neurotransmitters that they are targeting, right? In this multi-action group, it's a very different profile than the single action or SSRI group. When we move to the amygdala, I know there's a lot of bars and stars and things going on here, so I'm just going to walk through it by each emotion. So if we look at the negative emotion here, for negative stimuli, when they're correctly rejecting those lure stimuli, and we're looking at, again, basolateral amygdala is the darker bars, and the central nucleus of the amygdala are the lighter bars, and we have the responders in blue. We can see that the responders for negative stimuli are showing less activity compared to the non-responders for negative. But we see the opposite of that for positive, where they're actually showing greater activity during positive lure CRs compared to non-responders. So we thought this was really cool that we were seeing this kind of inverse valence-specific effect in the amygdala that depended on responder status. And when we look at that single versus multi-mechanism, here it seems like that effect is really driven by those taking multi-action mechanism antidepressants. So the multi-group, so taking the ones that are targeting different neurotransmitters, are showing that reduction in activity for negative and increased activity during the positive. And for neutral stimuli, I still don't really know how to interpret this, but we found that the activity in the central nucleus of the amygdala was greater than the BLA across the board. The BLA is more involved in emotional modulation of memory, so it might be why that's kind of ramped down in neutral, but I'm not too familiar with the central nucleus of the amygdala and why it might be more active for neutral. So any thoughts on that? Would love to get feedback on. But yeah, so that's what we found in the amygdala. So it seems like these profiles that we're finding in responders versus non-responders, or people taking different types of antidepressants, are valence specific, especially in the amygdala, where we're seeing this trade-off of negative and positive. Yeah.
There's no contrast against the ones that are the novels that are
the false alarms, or So yeah, these are all relative to novel. Yes, yes, for Zoom. Thank you. Yeah, so what is basically the question is like, what is it relative to? And so in all of this data, it's relative to the novel foils. Yeah, sorry, I just kind of skipped over that on the, yes, yeah. But yeah, the false alarms too is a whole nother story. Yeah, it's interesting, but it's more of what you would expect with that as well, with the valences and things. Okay. All right, so the last part of this section, we looked at functional connectivity between the hippocampus and the amygdala regions we've been talking about. And when we compare responders versus non-responders during those negative Luhr CRs, we find greater connectivity between the dentate CA3 and the BLA in particular, and not any of the other subregions of the hippocampus or the amygdala. Whereas when we look at that mechanism measure, so multi-action versus single action, we find a more broad pattern of greater connectivity in the multi-action group, where anything that's connected with the BLA from what we've looked at is greater connected. So the BLA is more connected with the hippocampus in general, as well as with the central nucleus of the amygdala. So, I think both of these findings tell us some different things about, in terms of responsiveness, that there might be, this might be a good biomarker to look at in terms of how the amygdala and the hippocampus are talking together in terms of being a responder or not. And then with the mechanism as well, I'm thinking about what are those neurotransmitter targets and how might that either broadly or more selectively change this connectivity between these regions. Okay, so to summarize this section of the talk, we've identified some interesting different profiles that are associated with mechanism of action of antidepressants, as well as response to treatment, as well as their interaction. And so we're hoping that looking at antidepressant use in this way can really help us better target and develop more targeted treatments and biomarkers that we can really kind of get treatments targeted towards people based on their symptoms and based on what we're seeing in terms of what is actually going wrong in their brain. So hopefully in our next study, we want to implement a pre-post design rather than just looking at people already taking antidepressants. There's a lot of complexity I kind of glossed over with, when you start antidepressants and a lot of other things. So we're hoping to do this before they start taking them and after to look at within subject change. We're collaborating with Renee Hen and Wei-Li Chang at Columbia on a project there, and then we're also starting one here soon, hopefully, as well. And then we really are interested in hopefully getting enough participants in those groups taking specific types of antidepressants to really explore those neurotransmitter profiles a little bit more selectively than just lumping them all together, which is what we've had to do so far. And then I mentioned briefly that there's lots of comorbid conditions with depression. And so we have a lot of data on this, and we're trying to apply it in like a transdiagnostic way a little bit more, and thinking about what symptoms do people have and how might that also be interacting with everything we're looking at. So hopefully we'll have more data on that soon in terms of trying to incorporate this very messy, heterogeneous group of people. Yeah. Okay. Any questions on that before I move on to the next part? Okay. So I'm going to switch gears a little bit, and we're going to switch to talking about aging, which is another condition where we see changes in memory, and in particular episodic memory. Aging has been associated with decline in episodic memory, although there's a lot of variability with age, so not everyone shows dysfunction. But on average, older adults, and I shouldn't even say older adults because it starts probably in your 30s and 40s, where we see this greater susceptibility to interference in memory based on that Luhr discrimination measure we've been talking about. So with age on these types of tasks, you start to see declines pretty early, whereas target recognition, you don't see as much decline. And so there's this bias away from pattern separation and towards pattern completion or generalizing across experiences that seems to happen with age across species. We see this in rodents and in humans as well. Along with those cognitive changes in the brain, there tends to be hyperactivity in the hippocampus, in particular in that dentate gyrus CA3 subfield. That happens with age, and it sort of peaks. Like if you're on this trajectory to Alzheimer's disease, it sort of peaks before like mild cognitive impairment, and then it starts to go down once you have full-blown Alzheimer's disease. But if you take levetiracetam, which is an anti-epileptic, it actually reduces hyperactivity in the hippocampus and improves memory performance. So there's something to this story of hyperactivity that happens in aging in the brain that you would think would be good, and maybe it's compensating for loss, and maybe it is, but it seems to kind of become maladaptive, almost like seizure activity, and how we kind of think about that. Some silver lining though, not all memory is lost or declines with age. Emotional memory tends to be relatively well preserved. Some studies that we did, again, using that same task, the emotional mnemonic discrimination task in older adults, we find both target recognition for emotional information and for lure discrimination was actually preserved in aging. So older adults tend to remember emotional information regardless of the gist or detail, which we found really interesting, since in young adults, we think that it's more adaptive to forget those details in emotional situations, right? And so it's interesting and kind of strange that older adults are still remembering that emotional information, even if it's the details that we might actually want to forget. So it's hard to interpret a little bit if it's good or bad, but this is kind of what we see on that same task. And when we look at the brain, we find signals, again, kind of consistent with this pattern separation signal during emotional trials, both for negative and positive information, when they're correctly rejecting those similar images. However, there's a large body of work suggesting that there's a positivity effect in aging. And we didn't really find evidence of that here. And so I've been curious of like, why is it sometimes there and it sometimes isn't? But the theory of why it might be there is based on the socio-emotional selectivity theory, where as you get older, you may become more aware of your limited remaining lifespan. And so you might prioritize emotion and well-being versus gaining knowledge that you tend to focus on more in younger years of your life. And so we wanted to dig into whether or not this positivity effect was a general feature of aging, or if there might be something more selective that's driving it. And so we thought about retirement as kind of this marker of maybe you're more aware of your limited remaining life because you're ending your workforce life, sort of starting on a new path. But again, you're very aware that it's like the last chapter of your life. And this has been associated with declines in cognition. As you get older, as you retire, you're doing different things, you're not as cognitively engaged. And so there's been some studies showing there's declines in cognition when you retire. And again, I mentioned they might be more aware of that limited remaining lifespan. Interestingly, the retirement age, which is 65 generally, although it's been going younger and younger in recent years, but 65 is also the age at which Alzheimer's disease risk begins to double every 5 years. And so there may be something to that in terms of retirement coinciding with this kind of very stark change in Alzheimer's risk going up. It's also a very understudied area of aging, and not a lot of people are looking at retirement. So we were very kind of interested to learn why. So we took the same strategy here and looked at individuals who were retired and still working. And we age matched them. So that was a big factor throughout both of the studies that I'm going to talk about today. So we collected a sample of working older adults and retired older adults across this lifespan of 55 and up. Or I guess in this study it was age 50 and up. I'm not going to go into too much detail because we're running out of time, but we collected that same task as well as a bunch of other questionnaires. And we also measured a positivity bias score. So that means how well did they remember positive stimuli compared to neutral stimuli, sort of this bias away from like this baseline of neutral. So when we look at those same measures of target recognition, We don't see any effects of group between the working and the retired older adults. But again, we see that lure discrimination seems to be a little bit more sensitive in picking up on these things, where negative and neutral memory seems to be worse in retired older adults, with no differences for positive lure discrimination. And when we calculated that positivity bias score within an individual and how they're remembering, each person had, positive relative to neutral memory, we found that the retired older adults showed a greater positivity bias compared to working older adults who almost, had the opposite, right? They have better neutral memory compared to positive. So this suggested to us that maybe the positivity bias is more selective in certain older adults who may be, again, potentially changing this mindset of like, I'm getting older, I want to focus on positive things. We didn't measure any of those things directly here, but that's sort of the thought of why it might be greater in retired, or again, worse in the working older adults. I'm going to skip over this part, but there's just some thoughts about whether or not this is a good thing or a bad thing in terms of, is it compensatory or dysfunctional? I'm happy to chat later about that if anyone's interested. But I want to jump to the imaging data for this same, it was a different group of older adults who were working and retired. And again, we include age as a covariate in all of these models because it's very much inherently linked to age if you're still working and retired, although we try to kind of have that overlap as much as possible. So when we look at the dentate gyrus CA3 subregions during those lure correct rejections, and then here also we plotted lure false alarms, you can see that the retired older adults in blue are showing this increased activity relative to the working older adults for neutral in both during lure correct rejections and false alarms, and then a greater signal during positive during the lure false alarms in those retired individuals. And when we look at connectivity between the dentate gyrus CA3 and the BLA of the amygdala, we find this interesting selective increased connectivity between these subregions, but for positive stimuli in those retired older adults. So there does seem to be something going on with positive stimuli in retirement, especially. It seems to be picking up on that hyperactivity that we've found previously to be dysfunctional. Maybe it is occurring more in these retired older adults, and maybe there's something to this positive memory, but it could be, again, kind of indicative of dysfunction or compensating for problems that might be going on. So we're still trying to work through sort of what it means in terms of these different types of trials as well, if it's lure correct rejections versus false alarms, and what that tells us about if that signal is compensatory in a positive way, or if it might be a sign or biomarker of dysfunction. Okay, so to summarize this portion of the talk, we found some evidence that the positivity bias may be more selective or stronger in retired older adults, and that this might correspond to greater hippocampal hyperactivity, as well as connectivity, depending on the valence of that information that we're looking at. And similarly, in this study, we're hoping to look longitudinally at individuals who are before they retire and track them over the course of retirement to see within subject change in some of these features. Lots of other complicating factors here, kind of similar to the antidepressant work. People are sometimes partially retired, so they're still working and they're kind of ramping down. So how does that impact all of this? And we had a small group in the last study I showed you, but it was only 13 people, so we decided to not look at it. But that is something that is very common and that would need to be examined, as well as the type of job that you are retiring from. So how cognitively engaging is it? Is it physically demanding? Is it stressful? Because that's going to have a big impact on whether or not retiring is maybe going to be beneficial for you or maybe detrimental. So we're sort of working through some of those other factors as well. And hopefully we'll be able to better tailor resources for people who are retiring to really adapt that to keep their brains and their bodies active in the appropriate way for them. Okay, so very quickly, just wanted to highlight the projects that I talked about today are these bolded boxes here. We have a lot of different other focused projects that sort of take into the account like the individual differences aspect, different links with like culture and Alzheimer's risk and how these things are all relating. So lots of other work that I couldn't talk about today that we're excited about, but it all sort of is rooted back to this emotional modulation of memory as a way to look at these different conditions and how what we remember and who we are as people and how that kind of takes you where you are today to like remember. Maybe you're going to be more biased towards remembering negative information or maybe positive information, really depending on that individual and their experiences. And just because I mentioned this briefly at the beginning, but we're really excited about incorporating these new or newer imaging techniques into our studies. So we're doing some locus coeruleus imaging, looking at, which is the primary source of norepinephrine in the brain. And so we're looking at that in correspondence to how norepinephrine, which is key in amygdala's modulation of the hippocampus, might be interacting here as like a biomarker potentially. We're also conducting MRS, which is, again, sort of a newer technique that's still being developed to try to measure neurogenesis in vivo in humans in the dentate gyrus. There's this signal at 1.28 ppm that has been associated with new neurons. And so we're really excited about this direction as well. We're collaborating with Craig Stark and Mariana Miletic-Savatic at BCM on some of that work. All right, so to summarize, we think the why of memory is really important in thinking about how our memory system works, especially episodic memory, and thinking about cognitively and neurobiologically, what kinds of profiles do we see when we look at different types of experiences in the hippocampus and the amygdala, what computations are supporting those things, and how are those changing in diseases like depression and aging. So hopefully the goal of developing more personalized interventions and better understanding this nuance that's happening in the medial temporal lobe will aid with that. That's the hope. However, it is very complicated because of all of these individual differences in our participants and human subjects that I think kind of get ignored a lot of times. And so we're really trying to be mindful of that in the data that we collect, in the individuals that we're testing, to really see how these types of individual differences like stress and culture and sex, all of these things kind of impact what we're finding. And so this why question really, I think, provides a greater context for how our memory system works and hopefully being able to target more specific dysfunctions in a wide array of disease beyond depression and aging as well. So with that, I'd like to thank my lab. It's listed here, and the studies I talked about their names are bolded here. Tried to mention them throughout, as well as our collaborators and our funding sources. And thank you all for listening as well.
Thanks, Stephanie. Great thought. Any questions?
Thank you for that wonderful talk and having me about here at UCLA. So a lot of the action to the co-behavioral effects is on the Lewer discrimination index side of things, and I know that measure intrinsically corrects for bias because you're looking at Lewer correct projection versus the Lewer false alarms ratio. But the bias itself could be an interesting thing in and of itself, but especially thinking of whether they have a tendency to do pattern separation and think of things that are a little bit different as being new, or pattern completion is a little different being old. And your candidates decide old, new, they don't get a choice to say same, similar, different. But I just wonder how you're thinking about it and look at sort of the bias of the people have a tendency to say old more or new more, and how that might be an interesting trait or musical difference to that they do depression or
Yeah. So the question, I mean, I was just going to bring up the, I won't repeat all of that, but basically like having a similar response in there as well. So we ask people to say old or new, and there's a lot of kind of this in between of tracking. Yeah, like if something is similar, how aware of it are they, and what is this kind of bias towards completing or separating? And yeah, I think that's a really important point. we chose to use the 2 response option to kind of simplify the story a little bit. But I know that, yeah, a lot of people use the similar in there as well. And I think you can capture different information with that. So yeah, we should try it in some of these populations that we haven't done. We've always just done the old new and tried to be consistent with it. But I think, yeah, with the behavioral data, we've definitely tried to look at like the correct rejections and the false alarms. The measures I talked about are corrected for response bias, but when you look at the like raw data, it's also interesting to see that like over aging. They've done a lot of work in that on other tasks. We haven't done it as much with the emotion task. So I think that would be something, yeah, we should definitely look at, see if there's anything maybe even more sensitive to some of these things, especially across valence as well. Yeah, yeah.
Yeah, the last slide you talked about the music.
Oh, yeah. Where did I have
I know it's a
that? Yeah.
you give us like one summary of.
Oh, you know, I don't know if I updated my extra slides with the music stuff, so I'll just have to tell you. But basically, we have one study published last year in J Neuro looking at, it wasn't this emotion task, but it was using the object version. So keeping that neutral, but having music played post-encoding. So they studied the objects, we played music for 10 minutes, let that arousal kind of go back to baseline, and then we tested their memory. And we found some really interesting things based on individuals' responses to the music. So it was all classical music to undergrads. So you can imagine, yeah, there was a very wide range of responsiveness. And when people showed really big changes, either in the positive direction, so more arousal, or actually decreased arousal, so extremes basically, we found differences in that gist versus detail trade-off. So if you had extreme changes due to the music, you showed better gist memory and worse detailed memory. But if you showed a moderate change, it was flipped. So we saw these like inverted V patterns. So if you had moderate increase in arousal, you showed better detailed memory, so the lure discrimination, but worse gist or target recognition. So that was super cool to us. We weren't originally planning to even look at it based on this change in arousal measure. We had like all of these groups of like negative music and positive music and familiarity and all of these manipulations. But the arousal and change in like that responsiveness seemed to be really tracking something about that quality and specificity of memory. So now we're doing some work with imaging. She's writing up her dissertation now and analyzing like 3 data sets with resting state music. Like, so we played music kind of in a similar paradigm, but preferred music. So that people selected, and we're looking at brain activity during that music, so during resting state when they're listening to the music, as well as impacts on like fMRI and on like the memory signals and things like that. So that's coming soon, hopefully in the next few months. Yeah.
I have a quick question about the signal quality in MTL and maybe the study use. What's the protocol? How do you ensure it?
Yeah, so I mean, we've been collecting these scans for a while now. We used to only collect like across just the medial temporal lobe for high resolution, but now we're actually able to get whole brain and have good quality data from the whole brain, although we still are focusing on the medial temporal lobe. But yeah, Craig Stark did a ton of like signal to noise ratio testing. We angle the box, and I was doing this like with in parallel with the hippocampus, so that seems to help. And then with the locus coeruleus stuff, it's still a work in progress, I think. We have like, we're running like 2 different types now. We have 3 different LC scans. So trying to figure out which one is actually the most sensitive. There seems to be more sensitivity in aging compared to young adults with specific types of LC scans. So there's a lot of different like nuance there that is tricky. And like how many slices you're getting from that region and the size of the box. It's like each one is very different too. So we're sort of still like kind of working on optimizing that at the Brain Mapping Center actually with Mary, and we're going to take a look at which one is going to be sort of the most sensitive to aging and to other biomarkers that we're looking at. Yeah. Yeah.
Can you have a question on Zoom?
Okay. Okay. Yeah, I can't like click on any of the Zoom stuff for some reason, but yeah.
But so there are no questions. Let's thank Stephanie. And
Thanks to all you who attended on Zoom.